Hi, welcome to our lecture on the respiratory system. In this chapter, we're going to take a look at how we take oxygen from the external environment and get that into our body, and how we take the CO2 we've been producing and get that out of our body back into the environment through these structures, which we refer to as lungs. So we're going to begin here with the four aspects of respiration. Respiration is going to have four parts, starting with ventilation. Ventilation actually has two different parts to it, inhalation and exhalation. So to inhale, obviously, is to bring air into our lungs. To exhale is to get rid of it. And these are governed primarily by Boyle's law, which states that pressure is inversely proportional to volume. Now, the simple way to kind of think about that is for inspiration. To take air and to actually pull that into our lungs, we're going to have to increase the size of our thoracic cavity. If we increase the volume of our thoracic cavity, increasing the volume of the lungs, because that parietal pleura is attached to the body wall and moves along with it, then we're going to decrease pressure and air will be drawn in. You can see here relaxation of the diaphragm causes it to go back up into a dome shape. Along with the use of some of our intercostal muscles, we can then actually reduce the volume of our thoracic cavity for exhalation. And you can see that there are alternative terms we can use, inspiration and expiration. So increase the volume of the thoracic cavity to inhale, decrease the volume of the thoracic cavity to exhale. Now, the other aspects of this are also going to be essential. They are really kind of the consequence of us being a multicellular organism. So I'm actually going to go ahead and do these kind of a little bit backwards here and begin with this thing down on the bottom. Oxygen utilization in the cell to form ATP is a process we refer to as cellular respiration. And so cellular respiration is an efficient process relative to forming ATP without oxygen that also requires mitochondria. And each of our cells needs to do this. That's why we have to have a respiratory system and a cardiovascular system to make sure that the oxygen gets to these cells. And because they are going to produce CO2 in this process, we have to be able to take that CO2 away. That's where the other two portions of this really come into play. And we'll jump back up here to the first one, external respiration. This is gas exchange between the alveoli and the blood. The alveoli are the thin sacs within the lungs, and then they have capillaries on them, and we need exchange of gases between those. So since the alveoli should be relatively high in oxygen, the oxygen will cross the alveoli and the capillary wall to get into the bloodstream, and the CO2, which is relatively high in the bloodstream, is going to cross the capillary wall and the alveoli so that we can then exhale that. The reason we kind of think of this as external respiration is because this area inside the alveoli where the air is really is still kind of outside of our body. That whole respiratory system is basically an invagination of the kind of wrapper on the outside of us. If we want to think about that, we might think about, say, our epidermis. But we can kind of follow that opening at our mouth and nose back through our throat, down our trachea, into our bronchi and into our alveoli, and until we cross those cell walls that are forming the alveoli to actually get into our body, we're still kind of outside. So we have this exchange between the outside and the inside. Then we're, of course, going to take that blood from our lungs, deliver that back to the left atrium, down to the left ventricle, and pump it out the aorta in the systemic circuit, eventually getting to the tissues that are actually going to need that oxygen. And here's where we'll have the process of internal respiration. Internal respiration will have that oxygen leaving the capillaries at these tissues and moving into the interstitial fluid between the cells and eventually into the cells. Of course, these cells have actively been undergoing cellular respiration. Therefore, they've produced a great deal of CO2. That's going to move into the blood so that that can return that back to the lungs so we can get rid of it via external respiration. So you can kind of get an idea that these processes are all related. We need to have all of them take place. And if we have problems with any one of them, we won't be able to form the HEP that we need to use to do basically everything inside of our body. So let's take a little bit of a look about how we do this with this respiratory system. We have two primary functional portions of the respiratory tract. We have what we call a conducting portion or conducting zone, which is basically the nasal cavities down to the bronchioles. We have portions of it here, and we can see that there are tubes. We have color changes here to go from our trachea to our primary bronchi, to our secondary bronchi, to our tertiary bronchi, to smaller bronchi, and eventually to bronchioles, which are going to have the alveoli branching off of them. All of these are just tubes for the transport of the air. They're not actively involved in exchange, meaning that they're not involved in that external respiration process. Therefore, they are just tubes for getting air from the outside to where the exchange process will take place. The respiratory portion then is what we refer to as respiratory bronchioles and the alveoli that arise from them. So you can see that this respiratory bronchiole is kind of the end of the tube system, and it directly has these little sacs, the alveoli, the singular form of that being alveolus, branching off of them. And of course we see the capillary networks on top of the alveoli, so we know that this is where that external respiration process takes place, where we take the oxygen from the alveoli and put it in our bloodstream, and the carbon dioxide from our bloodstream, and let that move into the alveoli so we can eventually exhale that. Now we do have another way in which we break up the respiratory system. So you can see here there's a little line going across, this dashed line right at the larynx or voice box. That little line in there is supposed to be the vocal cords. So at this point where the vocal cords are in our larynx, we're going to separate the upper respiratory tract from the lower respiratory tract. The upper respiratory tract is the nose, the nasal cavity, these paranasal sinuses that we see here in the frontal and sphenoid bone, and the pharynx, which is our throat. So when we think about having an upper respiratory tract infection, it's often something where we have an infection of our throat, some kind of sinus infection. Those are upper respiratory tract diseases. If we take a look at the paranasal sinuses for a little bit, we can kind of see why we might feel sinus pressure in various locations. There are paranasal sinuses in 4 different bones. We've previously seen those in the frontal bone, and then that yellow one in the back there is in the sphenoid bone. Those are the sphenoidal sinuses, basically sitting just below the sella turcica in the body of the sphenoid bone. We also have a bunch of small sinuses in the ethmoid bone, and these very large sinuses in the maxillary bone. So if you kind of feel near your upper teeth some sinus pressure, that's in your maxillary sinuses. You might feel a headache up in your forehead. That would be the frontal sinuses. If you feel pain kind of between your eyes, that's the ethmoidal sinuses. And if you have sinus pain that is so deep in the back of your head you're not really sure where it's coming from and it seems like it's coming from your brain, that's probably sinus pressure in your sphenoidal sinuses. Now here we can see the lower respiratory tract. So again, we go from our larynx and we work our way down through that into the trachea, into the bronchi, these large tubes, eventually to the bronchioles, the smallest tubes there. We're just going to kind of refer to alveoli and bronchioles as the lungs. This is our lower respiratory tract. So if we have a lower respiratory tract infection, that means that's down here actually in the lungs, potentially even involving the alveoli. It could be something like bronchitis, but it could be something involving, say, pneumonia, if this is a lower respiratory tract kind of infection. Now we want to think about the process of using all of these parts and getting air actually to where we need to get it to. So if we think about using this conducting portion, we're going to start with our inhalation at our nose, and this is where a lot of the gas conditioning takes place. So we obviously carry air, we can make sound, there are multiple functions of our respiratory system. This gas conditioning is kind of an important one. You can see here we have hairs that should keep you from inhaling large objects in your nose. We have these nasal conchae, which increase the surface area. They're all covered with that mucus-producing epithelium we have in this area. So we have the potential to filter out stuff, both large stuff with the hairs in the nose, and then in the mucus, kind of smaller particles. This gas conditioning process also involves adjusting temperature and humidity. We often think about this if we are in a very cold place in the winter, and in that kind of case, the air will be both cold and dry, and we need to warm it and moisten it up. Obviously, not every environment is the same. You might have an environment that is warm, where you don't really need to warm the air. In fact, you might actually cool it down slightly to body temperature as you breathe it in, but you still might moisten it if it's dry air, say of a warm desert. Obviously, in Ventura, our air is often moist, so adjusting the humidity isn't really as big of a deal. We're going to take a quick look at the tissues that are involved in the respiratory system, kind of using some review from our previous discussion of tissues, and we're going to recognize that the tissues are really based on the function of the particular portion of the respiratory system. So we're going to begin in the vestibule of the nose. Imagine starting to pick your nose. The external nares are where you stick your finger in, that portion of your nose that is kind of hanging off of your face rather than the deeper area where we have our nasal sinuses, but just this kind of triangular area out here, that's the vestibule. That area is lined by keratinized stratified squamous epithelium, the same tissue that forms the epidermis. And we actually know that this was going to be the case because when we looked in the nose before, we saw hairs in there just like we have coming off the outside of our body. So you can just follow your skin up into this first section of your nose, but that's not going to last forever. We also have in our oro and laryngopharynx, pharynx again being throat, and we name it based on different regions, we have the area that's filled with non-keratinized stratified squamous epithelium. So that's going to be this area of our throat back here and in this area here. And there's kind of a key reason for this. This is an area where we might have food and air. So we stick food in here and we have a mix of keratinized and non-keratinized stratified squamous epithelium within the mouth, depending on whether we're talking about the palate, the tongue, or the cheeks. And as we deliver that food back here, we need an area of our throat that still is going to protect us from the food that we're eating as we pass that down to our esophagus, which is down here back behind our larynx and trachea. So that area is a protective, stratified area, but it's deep inside, so it's going to be moist and it's not going to be the keratinized form. It's just the stratified squamous epithelium that is non-keratinized. What we've kind of skipped over is an area in here, in the nasal sinuses and in the nasopharynx. That's an area where we should only have air. We shouldn't have food going through there. So that kind of means we don't need that non-keratinized stratified squamous epithelium. We can actually get away with the ciliated pseudostratified columnar epithelium. And we're going to have that in our nasal cavity, our paranasal sinuses, our nasopharynx. We then have to go through that food and air area before we get down here into the lower part of the larynx and into our trachea, our bronchi, and the large bronchioles, areas where we should only have air being passed, but it's still part of that conducting portion where we want to have some kind of potential to filter out objects. Therefore, we need the mucus production that we associate with columnar epithelia. As you probably remember, columnar epithelia have goblet cells, those unicellular mucus glands, to make sure that this is a mucous membrane. Once we get all the way down deep inside the lung, we're going to have those little sacs branching off of the bronchioles, and those are going to be our alveoli, where gas exchange actually takes place with our capillaries. Of course, we're going to have simple squamous epithelium here because we know that that is our thinnest tissue, and we need to have those gases move across these cells via diffusion. Therefore, we need the thinnest possible tissue to facilitate that diffusion process. All right, so now that we've kind of got an idea of what's going on tissue-wise, we can just think about breathing in. We do have those external nares or nostrils on the outside, and we're going to breathe in through there, go through our nasal sinus. Again, we've got an area in here that is protective, and it's that keratinized stratified squamous, but we get into our mucus-filled filtering area in here, that ciliated pseudostratified columnar epithelium. We then pass out through another set of nostrils, which you can see in a skull back here behind this palate. And so there would be two openings into the kind of separated section of the nasal cavity. These are the internal nares, or the choanae. So the choanae is just an alternative term for internal nares. It's just an internal set of nostrils, which takes us into a single common passage back here, the nasopharynx. So just remember that there's a septum dividing our two nasal cavities through here. We have two tubes. We go to one tube as we go through those choanae. Okay, of course, that takes us into our throat, and we refer to our throat as the pharynx. I think people need to show some caution in pronunciation of this one. Sometimes people mispronounce pharynx and larynx, and when they do so, they often end up misspelling them. So it's inx in the end of this, and we can see that this is the throat area back here. Again, it's a mix of air only, and then areas behind our mouth and behind our larynx on the way down to the esophagus that could transport either food or air. We actually break up this pharynx into three different regions. This is a nice diagram to kind of give us an idea of what's going on there. We have the upper nasopharynx. This has the pharyngeal tonsil inside of it, or the adenoids. The oropharynx behind the oral cavity or mouth, and the laryngopharynx behind the larynx. So at least the names are really easy for those as far as the different portions of them go. Now, of course, our voice box is referred to as the larynx, and we do recognize that this larynx has a bunch of cartilages associated with it. We're going to take a look at the specific cartilages which are going to form this voice box. We can see this particularly in males, and we often refer to this as the Adam's apple because this is a large, kind of somewhat rigid cartilaginous structure here. It, of course, has to be relatively rigid to keep that area open so that we can pass air through it, and it does have some vocal cords inside of it that vibrate to give us our voice. We can see that in males in particular we have this laryngeal prominence here, which kind of sticks out and tends to be noticeable, although there is tremendous variation in the size and shape of the larynx, both within each sex and between different sexes. Therefore, there's not one particular appearance that you might have for a larynx. So as far as taking a look at the parts of the larynx, we have here the thyroid cartilage. Thyroid cartilage is the biggest piece. It does have that laryngeal prominence on the front that kind of sticks out there that we recognize as the Adam's apple. But this is the big anterior shield. It actually does not really exist on the posterior side, so we'll get that view a little bit later. And then down here we have a complete ring referred to as the cricoid cartilage. So those are the two main cartilages that we see on our larynx. Now it ends up that there's an important structure in between those where we can actually form an emergency airway. So it's important to recognize that a more permanent airway would be placed down lower in the trachea between the rings of tracheal cartilage, but an emergency airway can be done here in this cricothyroid ligament, or sometimes referred to as the cricothyroid membrane. You can even find texts that will recognize that portions of this are a membrane and portions are a ligament. For us, we'll treat it all as a single structure, and that means that if we find this laryngeal prominence on ourselves, we should be able to slide our fingers down and find a little divot in there, and that would be the cricothyroid ligament, which can be sliced through. There aren't any kind of major structures over the top of it, so there's going to be relatively little bleeding, and you can stick a tube in there if you have a blockage up above that that's preventing someone from breathing. I actually will include a short video on this in the modules section so you can take a look at this. All right, then we have the epiglottis. The epiglottis is important. It's going to change position during swallowing. It is the structure here that is composed not of hyaline cartilage as the rest of the cartilages are, but rather it is composed of elastic cartilage, meaning it will bend over. It can cover the glottis, which is the opening between the vocal cords, and prevent us from having anything that we swallow as far as food or drink going down through our larynx and into our trachea instead directing that food and drink down our esophagus. So the epiglottis we can see here sticking up. The hyoid bone up here is technically not part of the larynx, but you can see that it's attached via this thyrohyoid membrane to this thyroid cartilage. And because it's also attached to things like our skull via muscles, it's important for the manipulation of the larynx during swallowing. Now I'd like you to go ahead and take a quick time out, try to find your laryngeal prominence and put your finger on it and just make a swallowing motion. And what you'll probably notice when you do that is that your whole larynx is lifted up. Now it's that lifting up of the larynx that is going to actually fold that epiglottis over during the swallowing process, and when your larynx drops back down, the epiglottis, being made of elastic cartilage, will kind of flip back up into its open position, allowing you to breathe again. And here we can take a look at some other views of the larynx. So here's our anterior view we've looked at before with that epiglottis kind of popped up. We can see the epiglottis a little bit better from the back. It's going to have to fold down over structures in here. What I kind of want you to recognize is that this thyroid cartilage is a big shield. It's on the anterior side, but the back side of this larynx is largely open, but the cricoid cartilage is actually more robust on the back side. We do have other cartilages in here, the arytenoid, corniculate, and cuneiform cartilages, and these are going to be involved in the manipulation and movement of the vocal cords themselves. And we'll take a look at vocal cords now. We actually do recognize the existence of two different structures when we start talking about our vocal cords, what we sometimes call the true and false vocal cords, or what we're going to call here the vocal folds and the vestibular folds. So the vocal folds are these kind of white structures here. And this is going to be the boundaries for an area here referred to as the rima glottis. We're just going to refer to this as the glottis, but there is actually a couple definitions that go on here. Sometimes the glottis is referred to as this entire structure here, some of which obviously is not potentially open. The rima glottis is technically the open part of this that we're treating as the glottis. But the boundaries here are the vocal folds or true vocal cords, the ones that vibrate to create a voice. This vestibular fold out here obviously prevents any air from moving on the lateral side to these true vocal cords, basically making sure that this area is controlled, allowing air only to pass through this little area there. That way we can kind of decide how much air gets through here, and we can control what we're doing with our voice as we do that. Now this person here obviously would be incapable of actually making any sound. There's a lot of swelling and even some bleeding associated here with the vocal cords. So this is what laryngitis might look like, is you're going to have some kind of inflammation in there, and this swelling of those vocal cords would prevent them from vibrating as you pass air across them. If you've never had laryngitis before, you can try to speak and nothing really comes out. It's also generally going to tend to be somewhat painful, as we would expect when we have inflammation in an area like that. So here we can kind of get another idea of how that might look. Here we have the closed vocal folds, and you can see that we're not showing those vestibular folds, just the vocal folds. They are right next to each other. Here we see them opened up, and we can see what that would look like with those vestibular folds off to the side, again making sure that the air only passes in between the vocal folds or true vocal cords, so that we can close this and basically control how much air is flowing through there. Now once we do get air through there, it's going to move down into the trachea. So the common term we use for the trachea is the windpipe. That's this big tube right here, held open by C-shaped hyaline cartilage rings. So here we see one of those rings, and this is why we refer to it as a C. I know you might be looking at that and say, hey, from that particular view, it looks like a U. Well, if we turned it around, it might look like an N. It ends up that all those letters look kind of the same. But the idea is that it is closed here. We have all this protection on the anterior side, but the back side is open. And posterior to the trachea, we have the esophagus. So we need to allow for the distension of that esophagus. It needs to be able to bulge into this space where we would normally pass only air. during the swallowing process. So we have to have that open in the back where the trachea meets to the esophagus. We should actually take a look at one more thing here, and that would be this kind of production of mucus. We see a bunch of goblet cells in here producing a mucus layer, and we have the cilia have to be able to move that mucus up out of here. Now, this directly relates to the issues that people would have with breathing associated with cystic fibrosis. It ends up that what we can't see here is that there's also a little saline layer that is produced in here, which helps make that mucus more mobile so that it can actually be moved up towards the throat so that we can hawk up a loogie and then spit that out. And it ends up that the issue with cystic fibrosis through problems with chloride ion transport is the lack of production of a saline layer so that the mucus can't be moved out of here. It tends to get thick and build up, and that can, of course, inhibit the passage of air in the airways. So once we get down to the bottom of the trachea, we're going to split into the two primary bronchi. There's kind of a unique cartilaginous ring here called the carina to allow for that split to take place. And we're going to get our left primary bronchus heading over to our left lung, our right primary bronchus heading over to the right lung. Now, objects do tend to fall, if they get down in here, down the trachea and into the right primary bronchus because it's a steeper descent down into that, and the right bronchus is a little bit larger in diameter or wider. And that's really because the heart kind of takes up some of the space on the left side of our chest, meaning that it's a little bit tougher to get over to the left lung. It's pushed a little bit over to the left-hand side. And in addition to that, the left lung is a little bit smaller. Here we actually see that there's an earring in this person in the right primary bronchus where we would expect most objects to end up if they were accidentally inhaled. In this particular case, this person actually went to take a breath off of an inhaler, and the earring had fallen onto the inhaler. All right, so let's take a look at the lungs really quickly and kind of find out what those differences are between the left and the right lungs. The right lung has three lobes: the superior lobe, the middle lobe, and this inferior lobe. And we can kind of see here that the left lung only has a superior lobe and an inferior lobe. So there are two lungs on the left-hand side, three on the right. Now, it ends up that the lungs are relatively close to the same size, but of course that left lung is going to be a little bit smaller. Part of that has to do with some other features that are going on, and it's because of the heart impinging on that lung. We have what's referred to as a cardiac notch, which we can see from the anterior side of the lung. But if we look at this section of the lung, where we have kind of a concave area, this is going to have in it a cardiac impression. So this cardiac impression is basically a dent in this left lung where the heart is actually sitting. So there is a little bit of shape difference between the left and right lung beyond just the difference in lobe number. Now, we do also name the surfaces of the lung. We have a basal surface on the bottom, which is going to sit on the diaphragm. Most of the rest of the lung is the costal surface, all this that we see from the anterior side. And if you just trace your ribs, you can curve this all the way around to your back, about to where your spine is. So really, it's that whole kind of outer curvature, that lateral and anterior and posterior extent of the lung, which is the costal surface. Then we have that medial side, which is the mediastinal surface, which is going to face, obviously, the mediastinum. It's going to have the heart fitting in it, so we see our left lung here represented with that cardiac impression. That's going to be on the mediastinal surface. Now, it ends up that we do find something interesting going on there. There's an indentation referred to as the hilum, which is a concave section of an organ, whether it's the lung or the kidney or even a few of our other organs, where vessels or tubes go in and out. In this case, this is going to be an entry point for the bronchi, but also for the pulmonary vessels. We're going to have to be sending, obviously, our deoxygenated blood in through here, and that's going to be going in our pulmonary artery, and our pulmonary veins are going to exit from there. These other tubes are the bronchi. So that's the hilum. It's the concave area. We actually refer to that collection of vessels and tubes as the root of the lung. So if you were to have to remove a lung to do, say, a lung transplant, you would actually have to cut through that root there, through the pulmonary arteries, pulmonary veins, and through those bronchi. Same thing as if we were to remove a lung during a dissection. Now, once we get through all of the bronchi, we're going to eventually get down into some bronchioles. We can kind of get an idea here. We go through the trachea, past that carina, in this case into our left primary bronchus. We go into a secondary bronchus, into some tertiary bronchi. And you'll see as we head down into these smaller bronchi, we are progressively having less cartilage reinforcement. The primary bronchus has the same kind of C-shaped cartilaginous rings that we have in the trachea, but then we start to have a more patchy kind of network of cartilage supporting those smaller bronchi, and eventually when we get to the bronchioles, we don't have that cartilaginous support at all. These things instead are dominated by a smooth muscle layer, and then eventually give rise to those respiratory bronchioles, which have those alveoli on the end of them. So the key differences between bronchioles, these smaller tubes, and bronchi is the lack of cartilage and the switch to this smooth muscle dominating these. Of course, that can be related to some conditions that we have. Asthma, for example, is kind of muscular spasms of these bronchioles, which would be maybe of a less of an effect if we had cartilaginous support trying to keep those tubes open. Here we're going to take a look at the alveoli themselves. So we're all the way down to the alveoli. This is basically the end of the line for the respiratory system. This is the place where we're going to have that external respiration process, where oxygen, which is in these sacs, is going to cross those cells and get into the capillaries. And of course, the CO2 inside the capillaries is going to cross those cells to get into these sacs. There are a couple of different types of cells within our alveoli. We end up having things like alveolar macrophages, which can eat debris in there. So this is kind of our special white blood cell that's in the lungs. But we have our type 1 and type 2 pneumocytes, or sometimes referred to as alveolar cells. The type 1 pneumocytes are all of that simple squamous epithelium that is there to allow for diffusion. So all of these kind of hexagonal looking cells from this kind of top-down view, those are the type 1 pneumocytes. But we do also have some type 2 pneumocytes in here, and that's all these kind of weird looking cells that are scattered amongst these. And the type 2 pneumocytes do a particularly important function. They're going to secrete surfactant. Now you might ask, well, why do we need surfactant, and what in the heck is surfactant? We, of course, use surfactant all the time when we wash our clothes, when we wash our dishes, when we wash ourselves. This is a molecule which is capable of breaking up the attraction between water molecules. Water molecules, of course, are polar, and we're going to have the oxygen of one water sticking to the hydrogens of another water, because the oxygen is going to have a kind of net negative charge and the hydrogen ends are going to have a net positive charge. And this creates cohesion, or what we might sometimes refer to as surface tension, as that cohesion occurs on surfaces. And what would happen here with these wet parts of our lungs if we didn't have surfactant is that when we exhale, the side walls of the lungs would stick to each other, and then they wouldn't open back up, so we wouldn't be able to breathe as effectively. Well, the surfactant here is going to prevent that from happening so that we can have our alveoli kind of collapse somewhat during that exhalation process and open up during inhalation. And you're going to find out a little bit more about this as you kind of take into a look at something in the OpenStax textbook to figure out a little bit about the production of surfactant during fetal development and how this can cause problems for premature infants.